Good evening. My name is Saraah Zaheer. My research is within the Biological Sciences Department here at UA, and I work under Dr. Edward Burress. We are working to quantify and compare the feeding efficiencies of freshwater pufferfish and cichlids in snail consumption. To start on some background before going into my research, most fishes have a two-jaw system composed of oral and pharyngeal jaws. Their oral jaws are located closer to their mouths. These help them capture prey, and pharyngeal jaws are located near the backs of their throats, helping them process prey. In evolutionary ecology, the evolutionary arms race is the idea that in a predator-prey relationship, each organism is constantly evolving adaptations in response to the other. So, in our case, both pufferfish and cichlids have evolved highly specialized jaw systems in response to their prey, freshwater snails, who have evolved hard shells to protect their soft tissues. Pufferfish are unique in which they lack pharyngeal jaws and have a singular jaw system. They rely solely on their oral jaws, which are teeth that have been fused into what resembles a beak for both the capture and processing of their prey. Cichlids, on the other hand, employ the more common two-jaw system we see in most fishes, but have highly modified pharyngeal jaws that help them process their prey more efficiently. Some of these modifications include a mobile joint between each of the paired upper pharyngeal jaw bones and the neurocranium, and a muscular sling suspending the lower pharyngeal jaw from the neurocranium. My research is based off of a previous student's work here at UA. They tested the feeding performance of 3 different species of freshwater pufferfish, T. schutedeni, T. postulatus, and P. leiris, while eating 3 different species of snails, the Malaysian trumpet snail, the bladder snail, and the ram's horn snail. Each of these species has their own unique shell morphology, high, mid, and low spired, respectively. The previous research tested to see whether and how shell morphology influences predator feeding performance, and hypothesized that different shell morphologies would result in different processing times by the pufferfish. To better visualize shell morphology, we can reference the image on this slide. Whorls are complete 360-degree coils of a snail's spiral shell, and the spire is made up of earlier, smaller whorls. So when we're looking at the high-spired shell of the Malaysian trumpet snail, we can see that it resembles a tall, narrow cone, while the low-spired shell of the ram's horn snail is much flatter and rounder. The previous research found that size had a significant effect on feeding performance. So when we're looking at these 4 graphs on this slide, each is depicting the relationship between processing time and relative size of the fish to the snail. The one in the top left is doing so for all 3 pufferfish species, and the other 3 are doing the same thing but on a species-specific level. Here we can see that as size discrepancy between predator and prey increases, processing time decreases. This supports the intuitive idea that a bigger fish can process a snail faster compared to a smaller fish tasked with eating that same snail. The previous research also found that shell morphology and pufferfish species had a significant effect on processing time. Here we can see that low and high-spired shells incurred slower processing times in both the tetradon species, but had no effect on P. leiris. Moving on to my research. My research is still testing how shell morphology influences predator feeding performance, but this time we're working with cichlids who have that differing two-jaw system. We hypothesized that shell morphology would influence predator feeding performance, but also that the results would vary across the 2 different jaw systems. I worked with 3 different species of pufferfish, T. postulatus, T. lineatus, and T. imbu, and 2 species of cichlids, C. tesse and C. cametana. We're building upon the previous data, and our research question was, how does shell morphology influence feeding efficiency of pufferfish and cichlids, and do results vary across the 2 different jaw systems? My methodology for this project was fairly simple. I ran trials once a week, and during each trial, every fish was given one of each of the 3 snail species. I measured the length and width of each snail before feeding it to the fish, and processing time was defined as the time from the initial interaction between predator and prey to the moment that initial interaction between the predator and prey, so the moment that the fish grabbed onto the snail, to the moment that I could see that the fish had swallowed the snail, and this was measured using a stopwatch. As for my preliminary results, When we graph the relationship between processing time and relative size of the fish to the snail, we can see that as size discrepancy between predator and prey increases, processing time decreases, and this holds consistent with the previous research. However, when we're looking at this relationship on a species-specific level, the conclusions that we can draw from our results become a little less clear. For instance, when we look at the graph for the Cametana, for example, we observe positive slopes, which convey as size discrepancy between predator and prey increases, processing time does the same, which not only contradicts the previous research, but also the graph on this slide. However, this indicates the necessity of more trials to be run in the future of this project. But when we look at this on graphs that we had a bigger sample size for, like we do for the imbu, we can see that as size discrepancy between predator and prey increases, processing time decreases, and we see those familiar negative slopes. In this graph, we scaled processing time to account for the size advantage that bigger fish may have compared to smaller fish when eating these snails. Here we can see that low and high spired morphologies consistently encourage slower processing times across all species. When we're looking at the mid-spired morphology of the bladder snail, we can see that for all species excluding the Pustulatus, it was the easiest to eat. What's interesting here is that we can split this graph into two sections. The 3 species on the left are all pufferfish, and the 2 on the right are cichlids. Here we can see that the cichlids, no matter the shell morphology, had faster processing times when compared to the pufferfish, which conveyed that their 2-jaw system is outperforming the singular jaw system of the pufferfish. We can analyze this in terms of jaw morphology. So when in pufferfish, one big muscle called the adductor mandibuli is responsible for closing the beak. However, in cichlids, 11 small muscles operate the pharyngeal jaws. Out of these, one does most of the biting, but the others are able to articulate and rotate the jaw to accommodate a wider variety of shell orientations. So one possible explanation for the faster times that we are seeing in the cichlids is that pufferfish waste time trying to reorient the shell so that their beaks can crush it, whereas cichlids and their pharyngeal jaws with the 11 muscles don't have to do this. Before continuing, I'd like to circle back to my research question, which asked how shell morphology and differing jaw systems influence predator feeding performance. Our data definitely gets us closer to answering this question. We see that the ram's horn and trumpet snail morphologies encourage slower processing times, while the bladder snails seem to be the easiest to eat. As for the jaw system, we see that the cichlid pharyngeal jaws seem to outperform the oral jaws of the pufferfish. As for next steps,
do you foresee any issues with feeding the fish marine snails in the future iterations of this project, and why do you think they may occur?
Yeah, that's a good question. I do foresee some issues for feeding the pufferfish marine snails specifically, because before they got to our lab, these fish were raised in captivity, so we really don't know what they were being fed. So a lot of them come in with preferences against or for certain types of food. We can go back to this graph. On this slide, you can see that I don't have any data for bladder snails for the Pustulatus because it just refused to eat these snails. So my thinking is that when we're feeding these fish this unfamiliar shell morphology and food source, that they maybe won't recognize it or just prefer not to eat it.
So you mentioned the concept of the evolutionary arms race that gave rise to the specialized jaw systems, but since it's continuous, do you think there's any future counter adaptations from the snails?
Yeah, I think one plausible counter adaptation is the more complex shell morphologies that we're already seeing in marine snails. So because these shells are more complex, they take a longer period of time to consume, and in nature, time is a very valuable currency. So this is a really effective deterrent against the marine snails' predators. So because we're seeing this working for marine snails, that could be a plausible counter adaptation that the freshwater snails may have.
Was there something specific that made you choose cichlids as a comparative species?
Yeah, so aside from interest our lab has in these fish, cichlids made for a great comparative species because they are also natural predators of freshwater snails in the wild, but more than that, they have that deferring two-jaw system, which made for an interesting comparison between them and pufferfish.